Hi everyone, so I'm David Horn. So we work on African trypanosomes primarily. These are parasites that cause neglected tropical diseases. And our main focus is really molecular genetics, but we do a lot of cell biology and molecular biology as well.
Hi, I'm Melanie Ridgway. I'm a postdoc in David's lab. I joined a couple years ago after my PhD in Australia, and I specialize in parasites. So I've got experience in trypanosomes that David works on, and also malaria parasites.
Yeah, so this project, we spend quite a lot of our time running genetic screens. So we're interested in various aspects of how these parasites operate. And we tend to take an unbiased approach. So we have these high throughput genetic screening platforms, which allow us to really ask the question of which gene is involved in this process. In this particular case, so we've run quite a few of these screens historically, and in this particular case, we were interested in cell cycle. And a lot of people run individual screens on the individual cell lines, to look at individual genes and look at their function in cell cycle. The approach that we used here was actually to look at all 7,500 genes in the genome and ask how they impacted cell cycle when we depleted them individually.
And I guess after we'd run this large-scale screen on every gene, we then followed up a few of the interesting ones by looking at them in more detail.
So about 11 years ago now, we published our first screen, which was an RNA interference screen in African trypanosomes. And really, partly a technical reason to look at fitness, because really for that assay, all we had to do was determine whether cells died or survived after we depleted each individual protein. It was also extremely useful at the time to look at fitness because this was a way of prioritizing potential drug targets, because a lot of work we do in Dundee is really focused on developing drugs against these and related parasites. So fitness was a great way of prioritizing drug targets. The cells that when you depleted a protein, the cells died. This was potentially a good drug target. Cell cycle was a bit more complicated. So that's why there was a 10-year gap or more in between us running the fitness screen and the cell cycle screen. But the data are similarly interesting and useful because this can also help to prioritize drug targets and that just gives you insights into how the cells operate and function.
Yeah, I think the nice thing is, we're looking at the applied end of biology, looking for drug targets, ways to treat these diseases caused by the parasite, but also interested in just the fundamental biology questions of how the cell cycle is regulated for these parasites. So I guess when we're looking at a whole genome screen, like one of these, we do end up with a lot of hits, and you have to decide on the thresholds. So what is something that's important and what maybe isn't so important? So that's the first step. Second step is, I guess some genes had annotation and others didn't among those top hits. So the few that we followed up on in this paper weren't characterized before, so we didn't really know what they did before. So that was a power of looking at this whole genome cell cycle screen, was we could pick up genes that hadn't been associated with the cell cycle before.
And maybe I could add to that. I mean, I think trypanosomes are quite divergent from other commonly studied nucleated cells, such as our own cells, mammalian cells. And so actually a lot of novel mechanisms have been discovered by molecular biologists looking at trypanosomes. So we expected we might uncover some novel molecular mechanisms here. And we have found some candidates, but as Melanie said, it's going to require follow-up. So the screen really gives you some potential new hypothesis. It's a hypothesis generating kind of approach, and then we can pick the interesting areas that we can then follow up.
I feel like I just have a very broad answer to that. It just depends on what question you can think of, really. There's so many applications from my perspective. David, do you want to give a bit more specific?
But yeah, I mean, we've found genetic approaches, these high throughput genetic approaches, really powerful. The idea that you have a biological question that you're interested in. Maybe you don't have a clear hypothesis at the outset, but you have a clear question. To be able to sample every single gene and protein in the genome and in the proteome and ask, is this protein involved in this process, is incredibly powerful. You can imagine you could spend a lot of time going through 7,500 genes one by one, asking that question. So we found this approach really powerful, and it is versatile. So you can apply it to lots of different phenotypes. We've done a lot of work in drug resistance, for example. So you can run these screens to find out which genes are involved in drug resistance. So yeah, we will continue. We've used these screens quite a lot in the past, and we'll hopefully, and I'm sure, continue to do so.
I guess important thing to note with this interview is that a few of the other authors involved in this study aren't here. So this project had a pretty big bioinformatics component to it, and Melanie Tinti was the driver behind the bioinformatics analysis. And also Katrina Marcus, who's now based at the University of Glasgow, was involved in setting up the screen as well in the early days. So this has really been quite a big collaborative project, I suppose, within David's lab, and calling in a lot of different expertises as well. So cell biology people and bioinformatics. So it definitely extends beyond the skill set of one person.
I think the collaboration with Melanie Tinti has been incredibly important. I mean, we're dealing with increasingly, they're not huge data sets, but they are large, quite complex data sets that we deal with. And the bioinformatics input and contribution from Melanie has been fantastic. And we've been able to actually visualization of the data as well has been really useful. Melanie set up a database for us so that other people in the trypanosomes research community, and that's probably couple of hundred labs worldwide at least, can actually browse all these data sets and look at their favorite genes. So it's a case of access as well as actually allowing us to exploit the data, allowing other people to exploit the data as well.
In this group, it makes a lot of sense to have a lot of different expertises, because we do a lot of the whole genome screens. So it's important to have people that can do the wet lab work, so work on the bench, but also people that know how to analyze the data and visualize it really well. Yeah.
You know, I think I suppose, you know, we in one sense, so we need to seek people with particular expertise. In the case of bioinformatics, that's something I think we've become increasingly aware of, our need for expertise in bioinformatics. One other thing about Dundee is that there are great resources in terms of proteomics, for example. So I'm not really a biochemist background. Molecular genetics is really my background. So to have access to proteomics, for example, and other biochemistry expertise is really powerful. So proteomics is another example of a specialism that we can tap into in Dundee. One of the links that we have with the groups in Dundee is with the Wellcome Centre for Anti-Infectives Research. So we have a broader set of research labs that work on parasites primarily, and we're part of the Wellcome Centre for Anti-Infectives Research, which also is linked in with the Drug Discovery Unit in Dundee. So one of the things that we like to try and achieve through the synergy and critical mass is to really take our basic research projects and attempt to translate those into delivering preclinical candidates. So this project is probably not one of the best examples of that, but you can imagine having these large data sets means that if you're interested in a particular drug target, having that data already available means that you can learn more about that potential drug target and consider whether it's a promising target or not. So I think the message there is really about synergy and having experts with different interdisciplinary approaches to similar and related questions that can then impact human health.
So I guess a few of the hits that we were highlighting at the end of the paper, we're looking to look at those in a bit more detail in the immediate future. And then more broadly, I think David's lab will continue to use these whole genome screens to assess various other questions, probably. Yeah.
Yeah, there's one particular area that we're interested in, where it looks, we have some ideas. There's a new hypothesis around how the cell actually coordinates the segregation and replication of different parts of the cell, so different DNA components and different organelles. So that has to be coordinated in every cell when it replicates and divides. And so we have some new hypotheses around that question, and so we'll follow that particular question up. And we're really hoping that the community can exploit this resource for their research. There's quite a few labs that work on cell cycle control. We're actually not one of them that really focuses on cell cycle control, but now we have a new project in this area. So we will follow up on a particular question that we were interested in.